Right, limiting factors of photosynthesis. What this means is that the amount of photosynthesis that a plant can do is not always the same. Obviously, it can do more during the day than it can during the night. And we're going to look at 3 different factors that can affect significantly the amount of oxygen being produced by plants. So, these are 3 graphs that you might have a look of or that you might have to draw yourselves. And what we've got on the left axis of all of them is the amount of photosynthesis that is happening. This can be measured by the amount of oxygen bubbles being given off. Down here, we have got our x-axis, which is going to be the 3 different factors that we're talking about. And the 3 different factors are carbon dioxide, light, and temperature. So, what we see in this graph here is that the amount of photosynthesis is 0 when there is 0 carbon dioxide, 0 light, and 0 temperature. The amount of photosynthesis is at its maximum in carbon dioxide, when in the carbon dioxide graph, where you've got quite a lot of carbon dioxide, in light, where you've got quite a lot of light, and in temperature, where you've got the perfect temperature. And I'm going to explain these 3 graphs as we go along. Okay, carbon dioxide for first. We see in these 2 graphs that there's a very similar trend, that you get to a certain point, and after that certain point, the amount of photosynthesis that is occurring is the same. All right, this is my plateau. This is my level. This is where my carbon dioxide or my photosynthesis hits its absolute maximum. All right. The reason that is, I'm just going to draw a very quick plant cell. Okay, and in my plant cell, I've got to have a plant cell wall. Can't have a plant cell with a plant cell wall. And I've got my nucleus and my vacuole and stuff. But in my plant cell, I've also got chloroplasts. Chloroplasts are the machines inside the cell, the proteins inside the cell, that actually undergo photosynthesis. So when I've got a tiny little bit of carbon dioxide, maybe, or a tiny even little bit of light, maybe only one of my chloroplasts is able to function because there's only a certain amount of carbon dioxide for photosynthesis. As I increase the level of light or carbon dioxide, more of my chloroplasts are able to photosynthesize. Now let's just say I've got the amount of light and the amount of carbon dioxide necessary for all of my chloroplasts to be working as hard as possible. Each one of those is making as much glucose and as much oxygen as it possibly can. After that point, my carbon dioxide levels and my light levels just do not function. There's no point in increasing it. They're in excess. My chloroplasts are working as hard as possible. Even if I increase the carbon dioxide 1,000,000 times, they can't make any more glucose. And that is why we see this leveling off, because my chloroplasts are all functioning at their optimum rate, at their highest rate. Alright. Well, you say, why is it that my temperature graph doesn't show me the same trend? That's because temperature doesn't affect the chloroplasts in their function the same way it does carbon dioxide and light. What I'm going to do, I'm going to zoom in to a single chloroplast, alright? And inside this single chloroplast, I have got enzymes, alright? This is my enzyme who's going to undergo photosynthesis. This enzyme is the thing that's responsible for having my reaction happening. Now, what do we know about enzymes? We know that they're proteins, we know that the active site is very important, and we know that if the shape of that active site changes, then it can't do its job anymore. Well, if you increase the temperature to where it's very, very warm, eventually my proteins are going to start to denature. So it could be that if I come to this point, my protein, my enzyme, my active site, has changed its shape ever so slightly. Not enough to make it completely redundant, just enough to make it sure that it can do its job as well. If I increase my temperature even more, maybe that change is even greater. Alright? So this enzyme can still do a little bit of its function, but really it's not working as well as it possibly could. And when you get to a very high temperature, well, the shape has changed entirely. In fact, maybe even it's just a blob of protein somewhere, because it can't do its job anymore. Which is why we see this steady decrease as my temperature increases. So why is there this drop on the left-hand side? Well, just like every chemical reaction happening in the human body, temperature is important because it allows molecules to collide together. If you've got energy inside your cell, heat energy, then molecules can collide together. Now, I can have the best enzyme in the entire world, super effective, but if there's no energy in the system, if there's no molecules colliding, then it can't do its job. This enzyme can only work if it interacts with its substrate. So, let's just say I decrease the temperature a little bit. My enzymes have gone from being really, really happy to being actually quite sad, to being a bit ineffective. They've gone away from their optimum temperature. They cannot carry out the function that they're supposed to. Eventually, my temperature will be so low that over time, my enzymes can't work anymore, that there's no energy in the cell, that there's no energy there for my molecules to collide. So, my carbon dioxide and my light is a limiting factor because eventually my chloroplasts are all working as hard as possible. They're working at their highest rate. They can't work any harder. The reason temperature affects photosynthesis is because if you go away from the optimum temperature, proteins, enzymes can either denature or will have no energy, will slow down the rate of carrying out whatever function they have to do.